Hello students, I am Dr. Vaishali Bharambe, Professor of Anatomy. And today we are drawing the microscopic structure of bone, transverse section and longitudinal section. If you find histodiagrams difficult, then this is the series for you. Let's begin by taking a lead pencil and begin to draw as follows. There are three lamellae of bone now formed. I am going to color them to make you understand each of them clearly. Use dark brown and then yellow and blend them together as shown. Here you can see three lamellae, and in between lamellae can you see these tiny little gaps? These gaps are called lacunae. Now the next thing we do is we begin to communicate the lacunae with each other with the help of such fine lines which are called canaliculi. Okay, so in the next step now is to observe how the lacunae are communicating with each other through the canaliculi, and you're going to use your lead pencil to trace the canaliculi as shown. Let's now ask ourselves, what are these lacunae for and what are these canaliculi? These lacunae are spaces between the lamellae which are occupied by cells called osteocytes. Now please draw the osteocytes. As you can see, osteocytes are giving out cellular processes which are lying within the canaliculi, and with the help of these cellular processes, the osteocytes remain in contact with each other. Okay, so this is how nourishment crosses lamellae one after the other, reaching the distalmost lamellus. Draw the nuclei, color the cell. So this is the lamellar structure of bone, where there are lamellae. There are spaces in between which are called lacunae. The lacunae contain osteocytes. Osteocytes give out cellular processes which occupy the canaliculi. With this knowledge, we will go to the next part of our drawing, which is the drawing of an osteon, also called as a Haversian system. So let's draw the central canal called as the Haversian canal, and now all we are going to do is we are going to place the same lamellae that we saw in the concentric manner around this central canal. Observe. As we draw, we will leave tiny gaps between lamellae for our lacunae and osteocytes. So observe these three, 1, 2, 3, these are the lamellae. Okay, and this entire structure from one end to the other is called as a Haversian system or an osteon. Now let's begin to add the lacunae to this diagram. Between the lamellae are these tiny gaps called lacunae. Inside the lacunae, we know we will find osteocytes. Okay, so begin to draw, have some patience. We will now begin the next step of coloring the lamellae like I showed you earlier. So we will as usual use a brown shade distally and a yellow shade on the inner side of this each lamellar plate. Okay, have patience and keep drawing and blending in the yellow and the brown together till you are able to observe the lamellar structure clearly. Now the next step is for you to focus your attention on the central Haversian canal. Of course, under the microscope, this Haversian canal looks empty. But because we are trying to understand this, we are going to color it a little bit. We are going to color it red to make you remember that this structure actually contains blood vessels and nerves. Okay. Now what is left? Now what is left is drawing of such canaliculi. Keep on drawing canaliculi with patience. Okay. Remember, these canaliculi help nourishment in the Haversian canal to pass through canaliculi and go further and further distally to supply the distalmost lamellus. A simple white line extends around the osteon, indicating the end of an osteon. It's called as the cement line. So let's label this. So here you can see the lamellar structure of the bone. Since these are concentrically arranged around a central canal, we call them concentric lamellae. You can see in between lamellae lacunae containing osteocytes. Radiating out from them are fine canals called canaliculi, and of course in the center there is the Haversian canal. This whole structure is called as a Haversian system. If you have understood this, then drawing the transverse section of bone is going to be a cakewalk for you. Let's begin that by first drawing the chip of bone that we are going to draw. So a small wedge will be drawn by you. This is the outer surface of bone and this here is the inner surface of bone. So outer surface periosteum, inner surface endosteum. First let's draw that. Now we are going to focus on getting our lamellae in place. Of course we remember it's a lamellar structure. So begin to use your brown and yellow pencils to get the lamellae in place. Lamellae which are running on the outside beneath the periosteum are called external circumferential lamellae. Those which are running on the inside parallel to the endosteum are called as internal circumferential lamellae. So those which are on the outer side of the bone are called circumferential lamellae. Those on the outside are external, those on the inside are internal. Let's now begin to draw the osteons. So here you can see a number of osteons or Haversian systems containing a central canal and concentrically arranged lamellae all around. Okay. But besides this, what you are seeing are also small such bits of osteons. Okay. These are old osteons which are now being remodeled. Okay. They are called as interstitial lamellae. The ones which are drawn around a central canal are called concentric lamellae. Now let's begin to use our yellow color to further enhance the lamellar structure. Now begin to blend all these carefully together so that the yellow and the brown together look like one lamellus of bone. Now the next step is to define each lamellus clearly, defining a lamellus and keeping small spaces in between for the lacunae. Here you can see the central canal and the concentrically arranged lamellae all around it. These are the interstitial lamellae with tiny gaps in between them. As you can see, they are bits of osteons. They are not a complete osteon. Okay, they are bits of osteons undergoing remodeling. See how there are tiny gaps left in between for the lacunae containing the osteocytes. Now look at this and think what is left. Yes, we have to now begin to define our lacunae. Again, have patience, define the spaces, the tiny gaps between lamellae which are going to contain the osteocytes, define them well. And as you begin to define them, the whole structure begins to look like a TS of bone that you just saw under the microscope. Now take a look at it and ask yourself what is left. The canaliculi still need to be defined. Again, this is a little painstaking and slow process, but you have to go through it. It's a very commonly asked question and we must be able to represent the TS of bone well on paper. Let's now separately define our fibrous periosteum here and the endosteum here. Okay. We can also define the Haversian canals in the center. Remember, they will appear empty under the microscope. Here we are only defining it for our understanding. There, finally, you have the TS of a compact bone. Let's take a look at the labels just one more time. The outer periosteum, the inner endosteum, the outer circumferential lamellae external and inner circumferential lamellae internal. The concentric lamellae arranged around the Haversian canal and the small bits of osteons called interstitial lamellae which are undergoing remodeling which are present in between. Very simply, this is the TS of compact bone. With this knowledge, we go on to the last part which is LS of compact bone. To draw the LS, we have to first draw the Haversian canals. So these longitudinal are our Haversian canals and in between them what you see connecting the Haversian canals here is the Folkman's canal. So color the Haversian canals And now let's begin to draw the basic outline of lamellae. Okay. Remember, the lamellae this time run parallel to the longitudinal direction of Haversian canal. So all your lamellae will be running parallel to the Haversian canals. That is the basic idea. Between the lamellae, you will have to draw the fine spaces which are the lacunae containing osteocytes and the canaliculi communicating them with each other. The same thing will be repeated again at different places. Just remember that every lamellus you draw will be running parallel to the Haversian canal. Draw them. Color them using the same technique of brown on the periphery, yellow on the inside, and blending them together. In between lamellae, draw tiny spaces which are the lacunae, communicating with each other with the help of the canaliculi. Finally is your diagram of the Ellis of bone. You can see the Haversian canals. You can see the Folkman's canal connecting the two. You can see that there are lamellae running parallel to the length of Haversian canal. You can see tiny lacunae containing osteocytes and canaliculi extending outwards from it. Remember, the presence of osteocytes in lacunae is not assured because these bones chips are created by a lot of grinding. So most of the time the osteocytes fall off. But remember, the lacuna is the space where the osteocyte would be present. Okay. With this we complete the basic diagram drawing of the microscopic structure of bone, transverse section as well as the longitudinal section. For further details, you can watch my detailed lecture which will be released very soon. Thank you, students.